Good afternoon and good evening once more. My name is Vinolia and I'm the marketing coordinator for ISCA. I would like to take this opportunity to welcome each and every one of you who managed to be here today for this session. As you all know, we do have some housekeeping rules that we adhere to. Once you are in the session, you also have to abide by our rules. So I'm going to request for all of us, except for our speaker and myself as a host, to kindly mute ourselves to avoid any background noise or any disturbance. Can you kindly mute yourselves as we continue with the session? Thank you for doing that. Today we have our guest speaker, who is Dr. Nazia, who is from India as well. So Dr. Nazia Sultana is a NET JRF holder, a gold medalist, and a university rank holder. Her doctoral research titled Brand Positioning Practices of Banking Sector in India, a study of select banks from School of Management Studies, University of Hyderabad, has set a benchmark for all researchers in the area of study. She specializes in the area of marketing, and her other areas of interest include entrepreneurship development, human resource management, service marketing, and consumer behavior. She has won 8 best papers at international and national conferences and seminars and has published 24 articles in Scopus and UGC recognized international journals and journals of national repute. She has worked on ICSSR sponsored project under its Impress scheme. She has authored 4 books and another book is in the pipeline. She has contributed 15 book chapters in international and national edited volumes. She is currently research supervisor to 9 doctoral students, and she has organized conferences and several webinars and has been a resource person at various events. She has held various portfolios such as head chairman BOS at the Department of Commerce, UCW, student advisor as well, student counselor, and additional controller of examinations at the college level. Reading religion, philosophy, and writing poetry are a part of a leisure activities. I would like us to take this opportunity to formally welcome Dr. Nazia as she will be doing her presentation. Dr. Nazia, you are welcome on ISCA Research Webinar Series. And if I have missed anything, please feel free to take them through anything that I've missed. And thank you so much for being here.
Evening for me.
Sorry about the noise. Dr. Nazia, you can continue. Can you please, good people, can you please kindly mute yourselves because you are distracting the session, please. You can continue, Dr. Nazia.
Thank you, Vinolia. Good evening, everybody. I hope all of you are in good health and eager to learn something out of the session today. I have been associated with a lot of presentations and seminars which are on this topic of quantitative technique and analysis. And as a research supervisor, it's very essential that I guide my students as well with respect to how do they go about doing analysis of the data. What basically happens with most of the scholars around the world is when we have data with us, we are confused because there are so many tools which are available for the analysis of that data. So clarity related to what kind of a tool, what kind of a technique is applicable for what type of data is what is essential. And also not only scholars but also people who do research at a very high end, not only quantitative but also qualitative research, end up with a lot of confusion related to how do they go about analyzing and interpreting the data. No doubt there are a lot of softwares which are available currently, yet there are also hindrances with respect to how do you get the data and how do you make use of the data for the purpose of your research objectives. So what I will be taking you through in today's session is if you have data in hand, first of all, why do we do analysis of the data? And when you have data with you, what kind of an analysis can you do for that particular data? I would also be sharing a few of my own articles which I have published so that examples can be drawn from that for other scholars who are interested in doing studies which could be similar in nature. So if there are any questions in between the session, you can always put it in the chat box and I will take it up at the end of the session there. So let's begin. I hope my screen is visible to the audiences. So today's topic is quantitative data analysis and techniques, where we are going to concentrate on what is data, what is variable, and once we have data with us, how do we go about analyzing that particular data. And I'm thankful to African Institute for Supply Chain Research for having roped me in for this task of doing this presentation. I thank Ravikant also, who has been the mediator between me and between the institute there. So what I'll be taking you through in today's discussion is to do with this: what is meant by data, what is meant by variable, why is there a need for a scholar to analyze data, what kind of interrelationships are there when we do data analysis. This particular data which we talk about, we get through a lot of data collection tools. So when we have these data collection tools, if we make a mistake while designing the data collection tool, the data collection instrument, then our entire data analysis would go wrong and the interpretation also would be haphazard. So it is very essential when we talk about data analysis to actually talk about how did we get that data. So therefore, I will also be concentrating on measurement scales because the topic is quantitative. So my more concentration will be on quantitative rather than qualitative, where it has got descriptive and inferential statistics. And finally, I would close with what is hypothesis testing. So let's now see, any researcher, when you talk about in what way do I proceed with my research, we all have a research problem in our mind. When we take up a particular topic for study, there is a research problem for which we are trying to find a solution. And for that research problem, we start collecting data. We start gathering data from various sources. It could be sources which are first hand, where nobody else has collected data, which is called primary data. There are also sources where somebody else, it could be an institution or it could be other individuals who have collected data, and that is stored in some form which we are retrieving and using. That is called secondary data. So the data which is used by us to solve a research problem can be of two types. It can be primary, it can be secondary. Primary data is one where you talk about collecting data firsthand. That is, you are the person who is actually collecting data. This primary data you can collect through various ways. One can be in the form of questionnaire. You draft a questionnaire. Questionnaire is nothing but a series of questions, set of questions which you are chronologically putting it in order of your research objective, and then asking the respondents of your study to answer those questionnaires. So what you would do is, say for example, if I am doing a study on sustainable consumption and I want to find out what is it which is leading people to go for green products or organic products and end up indulging in a sustainable consumption behavior. So what I will be doing is I will draft a questionnaire and then administer that questionnaire to people who are engaging in a sustainable consumption behavior, who have got green buying behavior practices. This data, which I will get from such people who are called respondents, is called as a primary data. On the other hand, United Nations might have already collected some data on sustainable consumption because that is one of their SDGs, Sustainable Development Goals. So therefore, that particular data which United Nations have collected, I can retrieve from their database. That will be called as secondary data for me. So therefore, I have identified a research problem. What are the factors or what are the antecedents to sustainable consumption? I want to find out what are the antecedents to sustainable consumption. If I draft a questionnaire, administer it to the respondents, and want to find out what is it which is pushing people to indulge in a sustainable consumption behavior, that data will be called as primary data. On the other hand, if United Nations have gathered the data and I am retrieving that from their sources, from their reports, that will be called as secondary data. So what have I done here? Identified a research problem and got data for that particular research problem. Now this data which I have, which is primary and secondary, this I will have to take up to do analysis because data as such is raw in its form, because it is just to do with numbers. You cannot directly draw interpretation with numbers. You will have to apply some kinds of tools and techniques so that you do analysis for the data and then you reach towards the solution to your research problem. So this is what is the roadmap or the background for us to do data analysis as such. So you have a research problem, get data for the problem, do analysis of the data, draw interpretations, and then find a solution to the research problem. So in this backdrop, we understand that data analysis becomes very essential. So what is this data? Now, data is nothing but any kind of a measure which is obtained from some sample or from a population which is there. For this, we need to understand what is a sample and what is a population, which I'll come in a few minutes from now. Now, from this data set, you also will have to draw something called as a variable, which can be one of the characteristics of that particular data. So data is a measure on variables of any interest which is from the sample collected, whereas variable is the characteristic of the population of interest. Now this particular variable that we have got can be of two types. It can be a quantitative variable, it can be a qualitative variable. If you are able to quantify the variable in terms of any numbers, then it will be called as a quantitative variable. In case the variable is categorical in nature, where you are not able to directly quantify, but the quantification can be done by applying some scale or any other measure, then that will be called as a qualitative variable. So there are different techniques which are available for doing analysis of quantitative variables and qualitative variables. For example, in statistics, we talk about variables and we talk about attributes. Variable is nothing but that particular characteristic of the data which can be quantified. For example, if I talk about punctuality of participants joining this particular session, I can measure punctuality depending upon at what time they have joined, given a grace time of 5 minutes from the time session has begun. So this I am able to quantify directly. That is their punctuality, because number is available to me when they are joining. I have data with me, their login time is available for me, so I can quantify. But on the other hand, if I want to measure their attention, how much attention are they paying to what I am telling, then that attention is a qualitative variable. How do I measure their attention? So maybe Vinolia at the end of the session, when she takes a feedback about my presentation, and then if participants had got some questions which they want to get clarification upon, then I can measure attention, which is a qualitative variable which I cannot measure directly as I had measured punctuality. where their login times are available, and directly I am able to measure the variable punctuality, but the attribute attention I am not able to measure directly. I am depending upon some other process like a feedback or a question answer session in order to measure the attribute attention. So here, as a researcher, you need to understand whether your variables which you have in your data collection, are those variables directly quantifiable, or are they qualitative in nature, where you have to convert them in a measurement scale and then quantify those. There have been instances of research. For example, I have done one study which is to talk about store loyalty and store patronage. Store loyalty uh, and store patronage is to talk about what kind, how much of loyalty do customers show when they walk into a particular store, and what kind of a patronizing behavior do they have. So when you say store loyalty, this term loyalty actually is qualitative in nature. So you cannot as such directly quantify it. So therefore I use a measurement scale in order to get this particular attribute, which is loyalty. Similarly, patronage, patronizing behavior is also qualitative in nature, which I cannot measure directly. I will have to make use of some measurement scale in order to measure it. So your variables can be of two types, quantitative and qualitative. So depending upon the objective of your study, nature of the data which you are collecting, you will have to classify your variables into quantitative and qualitative. Variables which cannot be quantified, those variables which cannot be quantified, uh, such variables you will have to use some kind of a qualitative softwares, such as NVivo and others, which you make use of in order to do your analysis. But for quantitative data analysis, there are softwares which are available, such as Excel and SPSS and PLS, AMOS. All these are all SAS. These are all softwares which are readily available, where once you feed the data and understand the rules of the software, and define what kind of tests you want, the software will run the test for you, which is quantitative. Whereas for qualitative also, you have softwares like NVivo, which can run analysis on your qualitative data as well. So the clarity which a researcher needs to have is related to what kind of a data the researcher is collecting, whether it is quantitative or whether it is qualitative. Then the researcher also will have to understand, among the variables which they have with them, the data which they have with them, what is dependent and what is independent. So when I talk about um, factors influencing sustainable behavior of a consumer, then I will say price of a commodity, whether consumers' decision to buy green products is dependent on prices of these green products. So what I am trying to say there is, the prices of green products are influencing their sustainable behavior. So sustainable behavior is a dependent variable and prices is an independent variable, or sustainable behavior is the outcome variable and prices is the variable which is influencing variable, which is influencing the outcome. So you have to understand the variables which are dependent and which are independent in nature. So what is data and what is variable, you will have to first of all define in your while you do data collection itself. Identify what kind of variables you are collecting, whether it is qualitative and quantitative, and whether those and what kind of relationships they have among one another, that is, which is dependent and which is independent. Now once this is known to you, then you will have to understand what is population, what is a sample, and what is a parameter. Because when you are doing quantitative data analysis, we use these terms very frequently. Now, population is nothing but all the units which are eligible for the purpose of your study is called as a population. All the units which are eligible for the purpose of your study. For example, if I am doing a study on do discount influence men's shopping behavior, in case I want to do a study on, because the common perception is discounts influence women shopping behavior, but maybe I want to, do a study on whether discounts are influencing men's shopping behavior. So what happens there? I have now chosen a topic related to men's shopping behavior. So all the men in the world are eligible for the purpose of my study while they do shopping, but I can't study all of them. So that entire set of population, entire set of units which are eligible for the purpose of my study is called as population, because it is highly impossible for an individual researcher to do a study of the whole population unless and until the population is very small. Otherwise, the researchers usually choose few items which are representative of the population, which is called as a sample, and then do a study on that particular sample. You can always say that, it is not impossible for a researcher to do a study on the population. For example, if I'm doing a study on healthcare during COVID-19 and accessibility towards healthcare system by cancer patients. I want to specifically do a study only on cancer patients and whether they were able to have access to healthcare system during COVID-19. We are not talking about COVID patients, I'm talking about cancer patients. Now here, if I am taking my own state as my area of study, then I can calculate, I can get the data related to number of cancer patients in my state. So it is now easy for me because my population will be very small. Maybe, the number of cancer patients which I have, the number of cancer patients which I have could be only, could be only 20,000, for example. And it is possible for me to get data from 20,000. So I am doing a full population study. But then I am a small researcher and I can't do a study on full population, for example. So out of this 20,000, I will choose only 1,000. So that 1,000 is representing the entire population for me, which is called as my sample. So as a researcher, you have to understand whether you are doing a study on population or whether it's a sample study. Usually all our studies are based upon sample, except for few studies where your population is very small. For example, in psychological studies, you have few people whom you choose as subjects and you do a study only on that particular subject. So maybe 10 or 20, for example, you want to study behavioral changes in the parents of autistic children. And in my state, the number of autistic children are only 600, for example. I am able to reach all of them 600. So then all that 600 will be a part of my study. That means I am doing a population study. On the other hand, if I am doing a study on men's shopping behavior during discount sales, I'm not able to reach so many men across the world or in my state itself. Then I will choose a small sample, maybe 500 or 1,000, and I do a study which is representative of the entire population. Then my study will be a sample study. So as a researcher, first of all, define whether you are going to study all units who are eligible for the purpose of your study, or whether you're going to study only few units which are representative of your entire population, which is called as your sample. Do let me know in case you have any issues with my audio speed, speech style or anything. So let us not proceed with doubts in mind. Then we need to understand what is a parameter and what is a statistic. Now, when we are talking about this population and sample, population is all units eligible for the purpose of my study. Sample is few units drawn which are representative of the population. If I have information, if I have knowledge about the characteristics of all units of my population, then those characteristics of the population will be called as a parameter. As I said, 20,000 cancer patients, 600 autistic children. I have information about their social background, their family background. I have information about their economic background. I have information about all their, a set of all the characteristics. Then those characteristics related to the population, it will be called as a parameter. If a researcher has got knowledge about all the characteristics of the population, then the researcher can use something called as a parametric test. Parametric tests are those tests which are applied when a researcher has got an knowledge about characteristics of the population. I don't have knowledge about all the characteristics of the population. So when I said men's shopping behavior during discount sale, there are so many men in the world. I don't have information about all their characteristics. I have information only about the characteristics of few samples units which I have got. So the knowledge which I have about those characteristics of the sample units is called as sample statistic. So you call it as population parameter and sample statistic. Population parameter is nothing but knowledge about the characteristics of the entire population. Sample statistic is nothing but knowledge about the characteristics of the sample. If I have knowledge about the characteristics of the population, I can use a parametric test. If I do not have knowledge about the characteristics of the population, but I have knowledge about the characteristics of the sample, then I can use non-parametric. Non-parametric, that means I have no knowledge about the parameter, but I have knowledge about the sample. So then I can use a non-parametric test. This is where the researchers usually get confused. When do we use a parametric test and when do we use a non-parametric test? Knowledge about the characteristics of the population is known. You can always use a parametric test. That is the basic condition. There are other conditions also, but this is a first basic condition for using a parametric test. Knowledge about the characteristics of the population is not known, then you will have to use a non-parametric test. So this is, first we began with understanding what is data, what is variable. Under variable, what is quantitative, what is qualitative. Then we thought about what is dependent and independent. Then we came to what is meant by population, what is meant by sample. Then what is a parameter and what is a statistic. When can we use a parametric test and when can we use a non-parametric test. No, parametric tests can also be applied on samples. Parametric tests can also be applied on sample because you have knowledge about the characteristics of population. So obviously you would have knowledge about the characteristic of the sample also. So parametric tests can be applied on sample also. There's a doubt in the chat box. Is it that parametric test cannot be applied on sample? The moment you have knowledge about the population, it automatically entails that you have knowledge about the sample, because when you know everything about the population, automatically you know things about the sample also. So parametric tests you can apply even on sample also, because you have knowledge about the greater whole rather than the smaller thing that you have got. So parametric tests can be applied on sample as well because you have knowledge about the greater thing. Then why do we do analysis of the data? Because when we have data in hand, raw data does not convey anything on its own. We will have to apply some techniques in order to do analysis of the data and then draw interpretations from that. So why do we do analysis of the data? Firstly, we do analysis because we want to describe the data. What type of a data do we have? When I say we are collecting information about sustainable behavior, we are collecting information about accessibility to healthcare system or shopping behavior during discount sale, all these are my research problems and I want to describe the data. So I will do data analysis. Firstly, because I want to do a description of the data that I have. Maybe I want to describe my sample respondents. Maybe I want to describe their economic background, their social background, their family background, their demographic profile. So I am describing the data that I have got. So I do analysis when I do description of the data. Then I do analysis when I am trying to find out what are the relationships that are there between the different variables which are there. I want to draw a relationship between the variable. When I said sustainable consumption behavior and price of the commodity, is there any relationship between the two? So I want to draw whether there is a positive or a negative relationship between these two variables, price and behavior intention to purchase green products. So I am identifying what kind of a relationship exists between the two. And then I would also do analysis in order to compare the variables which are there. I want to draw comparison between the variables. Maybe if any one of you from South Africa is wanting to do a collaborative research with me from India, and then we do we collect data from your continent and this continent and countries, and then compare the data, compare the variables, and draw what kind of interrelationships exist. Then we are analyzing data by comparing the variables between countries, between different data sets, between different sample sets that we have got. So for comparing variables also, it will be essential to identify what kind of a differences lie between the variables, and then to forecast what kind of outcomes can be there, so that we can do some kind of a prediction for the research problem that we had got. Can we solve that particular, have we solved that research problem, have we given any kind of a solution to it, are there any forecasts which we have made related to the outcome. So we do data analysis related to that. There's a small fable which is there in Indian context, where we talk about data analysis. I will just share the fable with you. uh, there is this elephant, and there are 6 blind men, and these 6 blind men have to go and observe this elephant, and then do analysis of what they have observed. Observe in the sense, because they are blind, they cannot see, so they make use of their other senses, um, and then try to identify what it is. So one person feels the side and thinks that it's a wall because it's very hard. One person feels the tusk and thinks that it is a spear. One touches the trunk and thinks that it's a snake. Another feels the knee and thinks that it's a tree, then touches the ear and thinks that it's a fan, grasps the tail and thinks it's a rope, and they all have all these analysis which they have done. What happens with data that we have got is like this only. Blind men and the elephant fable. You may be given one data set, and the same data set may be given to another researcher. It is not universally applicable that what analysis you have done, the other researcher should also do similar analysis. It all depends upon the objective, the hypothesis, the kind of techniques that you're using, that your analysis and the other person's analysis may always differ. So it is not essential that data analysis is universally applicable across researchers who have done research on similar topics. We must have seen, when we do review of literature, we come across, a lot of researchers who might have applied, one researcher might have applied one technique, another researcher might have applied another technique. So therefore they have drawn different inferences, related to different techniques that they have applied. So it is not necessary that we all should be applying same technique all the time. It all depends upon the purpose of your study. It depends upon the hypothesis that you have got, that you do analysis depending upon what is your purpose. Why am I doing the analysis? Say when I said, is price impacting sustainable behavior? Is, for example, crowdedness. If the store is very crowded, do I engage in a patronizing behavior? Maybe if the store is not, if the store is very crowded, I may not like to go to the store, and my patronizing behavior may be very low. So what happens there? I am analyzing in one sense, and another person may be analyzing in another sense. Somebody else may be wanting to go to a retail store when it is more crowded because they feel the sense of satisfaction while shopping, while others may be wanting the store to be less crowded to have a more enjoyable shopping experience. So then the interpretation that I will draw will be different from what the other person must have drawn. So analysis of research is just like this Indian fable, where you talk about, different people drawing analysis for the data in different ways. But if two researchers have got same data set and are applying same technique, then definitely the research results should be same. If only that the data sets are different, if only that their data sets are same but they are using different techniques, then results can be different. But if data set is same, technique is also same, then result also should be definitely same. But if techniques are different, always the results are also different. So it is just analysis of the data is just like this Indian fable. I've done a study on entrepreneurship and incubators, where I was doing analysis for my own data related to 10 incubators who were in my state, and another 10 incubators who were outside my state. So what happened at that point of time was, when I did analysis, I found that, the tool, the technique which I'm applying for the incubators who were in my state, I was not able to apply the same to the incubators which were there in the other state, because there was missing data. Some data was not applicable for them. So therefore I had to make changes in the technique. I have to change the technique and then do analysis in a different way. So that way, what happens, data analysis always differs from person to person, depending upon what is the purpose of your study and depending upon what kind of an objective are you trying to attain by using that particular technique. So it is just like this blind man and the elephant fable which is there. Now, when you do data analysis, first of all, there are some interrelationships that you draw. You need to understand that you start your, maybe some of your previous sessions on research studies must have addressed to you how do you go about identifying a research problem, how do you frame research hypothesis, how do you do literature survey, because I am entrusted with the task of data analysis. So I assume that these must have already been done to you. So now when you do data analysis, everything has got a linkage backwards. Unless I know what has happened in the literature, unless I have a thorough grounding on my theory, I will not be able to do a complete justice to my analysis. That's why it is very essential that I have to do a complete literature survey. Most probably, maximum number of studies that must have been done in my field. For example, when I was doing my thesis on brand positioning, um, I started right from 1965. Al Rice and Jack Trout, who were the pioneers on brand positioning. From there onwards, till the time I submitted my thesis, almost all the studies across the world on brand positioning, which were actually there in the public domain, I have read all of them, because I can't have access to those data which is in the private domain of people, because of accessibility. But whatever is in the public domain, I have read all that, so that I have a complete understanding of all variables which I can cull out from the literature, I can put in my study. And then I also have complete knowledge about what are the gaps in their studies, so that I can put it as an objective in my study and further take the process of that particular research topic. So therefore, when you are doing methodology and data analysis and outcomes, you will talk about the source of data and what kind of techniques are you applying, what kind of hypothesis have you drawn, and how are you drawing the interpretation for that. So everything has got a linkage to, what you've done previously. Data analysis also has got linkage to what was your research problem, what are your objectives, and what kind of a survey have you done to your literature. Then, as I said, data analysis is dependent on how you get data. So how you get data is based upon different types of data collection tools. You can get data through a questionnaire. I have series of questions in my mind. I draft the questions in the order of their priorities, and then I ask respondents to answer those questions. That is the simplest thing which most of us across the world use. Sometimes my respondents are um, uh, not aware of the language, not educated to answer my questionnaire. So then I will go by something called as a schedule, which is an enumerator schedule. So I draft a schedule and I ask questions, and as they keep answering, I draft a transcript. And then later on from the transcript, I will cull out what were the responses. Or I can do an interview method where, as the interview proceeds, it can be structured where I have questions in my mind, or it can be unstructured. Depending on what kind of responses I get from the person, I keep on asking the questions. So interview method. It can be observation method also, because we are talking about social sciences. So you can also do observation. For example, I want to observe shopping behavior of parents when accompanied by their children. So when children go along shopping, is there a variation in my shopping behavior and I don't take my children along? So therefore, that's, if I ask apparently questions, they become conscious of their behavior. So I do not want to ask questions. I just want to observe whether children are influencing shopping behavior of their parents. So then my study will be observational study. These are applicable in social sciences, but you talk about pure sciences like physical sciences, biological sciences, medical sciences and all. Then you have lab experiments which you do, which is mostly absent in case of social sciences except for psychology. But other science we do not, because society is our lab and we do research in society. So therefore we take data depending on all these scales. Why this becomes essential? Because when we do data analysis, measurement scale is very essential for us. How are we getting data? If we are using any one of these scales, based upon what scale we are using, our test also will be different. First scale that we can use is called as a nominal data. Nominal data, as the very word says, nominal, it is only data where you can only do description. You cannot run any analysis on nominal data. You can only describe the data. For example, gender, ethnicity, these things, or income levels, education levels of people. I am only trying to understand, I'm only trying to understand name of the person, gender of the person, ethnicity of the person, where it is only for naming purpose, where I'm describing the data. These scales will be called as nominal scales. For such scales, there is no quantitative technique which you can apply. You are only describing. For example, you will describe number of men in my data, number of men respondents, number of women respondents. So male and female respondents. So what I will do, I only take mean. So percentage out of 100%. How many men were there in my study, how many women were there in my study. So what am I doing? Only describing through mean, percentages and frequencies. That is your nominal data. Second type of data that you have is called ordinal data. Ordinal data is one where you're classifying the data and you're also able to give ranking to that particular data. You talk about income level, you talk about educational background, you talk about the number of members in the family. All these are all called as ordinal scales, where you're able to classify the data and rank that particular data. So for example, you talk about I am thinking about does income impact shopping behavior, for example, or people belong to low income groups were not able to survive during COVID. So when I have such studies which are directly related to income levels or employment levels of people and academic levels, educational background of people, then I am classifying and giving rank to that particular thing. So here also I'm not applying high end quantitative techniques. I am only classifying and doing ranking for the data. So then I am using an ordinal scale. Then you can use an interval scale, where you talk about predetermined equal intervals will be there, and those predetermined equal intervals are used, and then you are applying some kind of a quantitative technique there, a little above your classification and description. The third scale, which is called as your interval scale, which will be there, as I gave examples of IQ level, achievement, aptitude and all, those will be a part of your interval scale. Most of the time, we as researchers in social sciences, we use the ratio scale, where we take up on a scale of 1 to 5, which is a Likert scale popularly used, or on a scale of 1 to 7, or a maximum Thrusten scale, which is a scale of 1 to 9, where we, for example, after the session, if Vinolia wants to take a feedback about the engagement or attention, or the degree of liking of the participants about this particular session, she may put it on a scale of 1 to 5 and then say that excellent to poor, or engaging to not engaging, so that satisfied to dissatisfied. So that particular scale will be called as your ratio scale. So why this is essential for data analysis is, unless I know what kind of a data I got and what kind of a scale I used in my questionnaire, I will not be able to apply the technique. So for me to apply a particular technique, first, and when I'm drafting my questionnaire, I have to be well aware about what kind of a scale I am using, because if I have not used the scale, I will not be able to use the quantitative technique. That is why this becomes very essential for doing quantitative data analysis. Understanding what kind of a measurement scale have you used while you were collecting data. It so happens that most of the time researchers come to us after they have collected data, and they tell us that why is it that I'm not able to apply the technique, because you have not collected data which is related to that particular scale. So therefore, that scale becomes very essential. And most of the time, once you have drafted the scale, how do you know that you're proceeding in the right direction or not? So that you can know by testing that particular scale. So you can test the scale for two things. You can test it for reliability and you can test it for validity. Reliability will tell you whether the scale is consistent, whether the scale is stable, and validity will tell you about whether the scale is accurate or not. So therefore you have to talk about reliability and validity of the scale. Most of the time, we if you've done some papers and all the audiences present here, we are running cronbach alpha in order to know the reliability of our test. And we also check validity by applying the validity tests which are there in our softwares which are available to us. So these are some of the tests which are applicable for testing whether your questionnaire is going to get you reliable data, whether the data is going to be accurate or not. So you can run a reliability test. It can either be cronbach, it can be a split half, it can be inter-item consistency. Any one of these tests you can apply. Maybe I'll show you one questionnaire, and then I'll tell you, whether what kind of a tests reliability score can be applied. So these basically will tell you, about whether whether what you have done is correct or not. So if you look at this particular questionnaire, which is there, this was a study which I was telling about sustainable consumption I was doing on Indian consumers. And see, first question I said name, that is nominal, gender, male, female, others, nominal. So now then I talk about occupation, annual income, I talk about age, number of members in the family, education, background, up to question 3 to 7. These are all ordinal scale. And then I have questions where you talk about, ranking being given. See, which of the following types of green eco-friendly products do you use? Are you satisfied? There I'm putting it on a scale. Very satisfied, satisfied, neutral, dissatisfied, very dissatisfied. Then you can also have questions which can be dichotomous, where you have yes or no. So these are called two options. When you give two options to people, dichotomous, yes or no. So here yes or no questions are there. Then the remaining questions that I have are all on a scale of 1 to 5, where I have kept 1 as strongly agree, 2 as agree, 3 as neutral, 4 as disagree, 5 as strongly disagree. You can reverse this also. It all depends upon what kind of analysis you do. There's no hard and fast rule for this. So here I am listing the variables which are there: personal stimuli, environmental stimuli, social stimuli, knowledge stimuli. So like that I am listing, and then I want to find out of all this which is going to yield me the result or not. And then once I have done this, I am running cronbach. See, I want to test whether this particular questionnaire is going to give me reliable results or not. So then I have run a cronbach alpha test here, which is a reliability test. And then the number of items which were there in my test were 47. And then the score is 6.12. Any score which is above 60 is supposed to be a reliable score. Nowadays, most of the scholars are using a lot of items in their questionnaire and getting a high score also, up to 90-97. I also got for some. This way, in case you are using frequencies and percentages, this will be your descriptive statistics. You are using descriptive statistics to do analysis of your data. So I am just noting the number here and the percentage here. I'm not applying any other high-end tool here. It is only simple percentages, because these are all to do with descriptive statistics that I'm using. Later on, as I proceed, then I have my hypothesis, and then I'm applying different types of tests which are there. So let's get back to the PPT that was there. So questionnaire you have to draft and use any kind of a scale, and then check whether the questionnaire is valid or not valid questionnaire which was there. Yes, most of the researchers are recommending moving away from cronbach to split half, inter-item consistency. Because split half, inter-item consistency, these are giving you a higher alpha score, coefficient score. That's why most of them are recommending. I personally also recommend that, you know, split half is much better. The test retest is much better when compared to cronbach alpha. That is also my personal observation, which I have got, because the reliability score is very slightly when you apply cronbach and when you apply the other reliability tests which are there. Now, when you do analysis of the data, depending upon what type of data, you can have analysis which is qualitative and quantitative. We are not talking about qualitative analysis because we are focusing only on quantitative. In quantitative, you have two types, descriptive and inferential. And these two, you can make use of the software also in order to do your analysis. You can do it manually also, but it is very time consuming, and most of us are tech savvy and used to some softwares which we use. So most of us prefer to use computer softwares which are there. Descriptive statistics, as the very word says, descriptive. We are making use only of simple mean, median, mode in order to describe the data. We are not doing any interrelationships or any comparisons. We are only describing the data. So hence the statistics which are used are called as descriptive statistics. On the other hand, if we are using statistics in order to draw inferences, then we are using inferential statistics. That is, draw some kind of an interpretation, relationships, comparisons, find out the impact of dependent on independent variable, etc. Then we are using inferential statistics, which is there, right? So descriptive and inferential statistics. So measures of central tendencies, measures of dispersion is used for descriptive statistics. And for inferential statistics, depending upon whether you have knowledge about the parameter or whether you do not have knowledge about the parameter, you make use of parametric and non-parametric tests. So you can use software such as SPSS, SAS, Stata, AMOS, PLS, all these are all there for structured equations. So those also can be made use of. Yes, you can mix all types of questionnaire scales in the same questionnaire. You can mix all types of questions and all types of scales in the same questionnaire, because the analysis that you will do is not for the complete questionnaire. You choose the variables which are appearing on certain cells in your Excel sheet or in your SPSS sheet, and only on that you will run. So therefore, if you are using a dichotomous scale, for example, yes and no, I can use a chi-square there. On the other hand, if I'm using a 5-point scale, I can use an ANOVA there. So therefore, in my questionnaire, it is not necessary that I should have one scale only that I'm using. I can make use of different scales in my questionnaire. You can make use of a differential scale where you have only no extremes, and in between everything, you're not listing everything in between. That's a semantic differential scale. You can also have listing of every item in the scale like a Likert scale. So you can also have a dichotomous yes or no type of a question. So all scales, mixture of scales can be used in the same questionnaire, depending upon for which question you want which type of analysis. Then you apply that data only for that particular analysis which will be there, right? So you can make use of these softwares also. I'll just list some kind of tests which can be there. These are some of the non-parametric tests when you do not have knowledge about characteristics of the population. Then you can make use of these tests. I am leaving the presentation with the institute, so you all can that they can share it with you, and you can have an understanding of, you know, what type of a test can be used by the researcher when you have such type of data with you. So when do you use, for example, when you have chi-square, which is a non-parametric test, you can use this when you have nominal data for one sample and or to a more dependent sample. And what do you test? You can test for independence of variables. You can use Cochrane, you can use Fishers, you can use sign test, median test, Mann-Whiteley test, Cruxell-Wallis, Friedman and ANOVA, Kolmogorov, Smirnov test. All these are all non-parametric tests. Conditions are listed when you can use and what function will the test perform in case you use this particular thing. It all depends upon, you know, what is the objective of your study, what is the purpose of your study, what is it that you want to attain. For example, if somebody wants to find out bivariate correlation, then you can directly go for Pearson or Spearman correlation and then, you know, run your correlation test, for those two variables and bivariate correlation coefficient you can draw and do an analysis of that particular data. So it all depends upon what is the objective and the purpose that you have. And these are different other types of tests that you have which you can use depending upon how many variables which you have and whether the variable is categorical variable or a continuous variable. So you see you have one variable, you have more than one to two variables, and then these variables are in terms of categories or these variables are in terms of a continuous scale. Then depending upon how many variables you have, you can use any one of these types of tests which can be there, because the time is only 1 hour, so I'm just running through what kind of tests can be used, and I'll show you how these tests are actually being used. So see, if you somebody was asking, can we apply a parametric test even for a sample? So this is where you can apply depending upon variables which you have got, whether you use a non-parametric test. If you don't use this, you can also use an independent t-test. Wilcoxon test, if you don't use, then you can use a paired t-test. So there is always an option which is available. It's not that when you have to use one, you have to use that only. It all depends upon what type of a data you have got. Say for example, correlation you want to draw, and you have variables which are bivariate in nature. In case you don't have knowledge about the population parameters, then you are running a Spearman. In case you have knowledge about the population parameter, same test which was Spearman is now Pearson, because you have knowledge about the population parameter. So it all depends upon um, what kind of a data you've got. And once all these tests, you have to again go back to what was your hypothesis, and then that particular test you have applied, and then you draw an interpretation for that hypothesis. So let me take you to one paper, and then I will try to show you what were the objectives, and then how did we go about this paper is related to crowdedness, and yeah, crowdedness on patronage and customer decision. This is along with other two co-authors which I have. Now here what we are trying to find out is whether crowdedness, number of people in a particular shopping store, have got any positive effect on my patronizing behavior, consumers patronizing behavior. So I have chosen some hypermarkets in India which are offering everyday low prices, and then I want to find out this particular thing. So what I have done here, uh, to understand this, I have this data with me. So I have data which is related to male, female. So I have simply done frequencies and percentages, age of the respondents, frequencies and percentages, education, frequencies and percentages. So simple description, because these are all data with which I cannot do much, but only describe. Then I have data where I am talking about, what kind of correlations exists. So then I am running correlation here. So Pearson's correlation I'm running here, where I know that, crowdedness and patronizing behavior has got this much of a correlation coefficient. So correlation I have run between these two variables, bivariate correlation between crowdedness and patronizing behavior. This was one of my hypotheses, where I was trying to find out is there is no significant relationship between crowdedness and patronizing behavior. So then to test that hypothesis, I have run a Pearson correlation test, and then I have got this particular score of 0.69, saying that there is a positive correlation between crowdedness and patronizing behavior. Then I have also run a regression in order to find out the impact of crowdedness on store patronizing behavior. So there I got all these scores. See, this R in your ANOVA summary will tell you about the degree of correlation. R adjusted and R square are related to adjustments which are made after adjusting for your errors, sampling errors and missing data errors which you have got. Then this is your final significance value which you have got, or this is a calculated value of t that you have got. So when usually as researchers, we are taking this significance value and comparing with the p value in order to find out whether we are going to accept or reject the particular hypothesis which is there, right? So this is one paper that was there. Then one more I can show. This is another one where we are talking about, running a chi-square test, running a regression also, where we have drawn a regression equation. So this y is the dependent variable, x1, x2, x3, x4, 5 and 6 are independent variables. All these together, whether they have got any kind of an impact on your dependent variable. This is what we wanted to know. So basically we are trying to find out whether marketing strategies adopted by companies are having any impact on the environmental claims made by these products which are there. So we talk about informating advertisements, green content. Here in this paper, we sort of did recollection of data. We collected data twice because our R score was very low. It was only 0.139, which is a very low score, meaning that there were some variables which you have not considered because the correlation with which we were not getting the correlation between the variables. So unless you have a R score which is higher than 0.6 or closer to 0.6, then you can say that you have taken into consideration all variables. Because this was very low, so we did a collection of data once again in order to again do this particular test. So this way you can find out. So here you see we got this particular significance value. This is one way which you can run on over and then draw conclusions and put suggestions for your study. So this one type of a test again which you can have. Similarly, different depending upon whatever is your purpose of your study, as I said, you can have different types of tests which you can apply. Is it advisable? Yeah, there's no limit on a number of hypotheses that you can have for your research. It depends upon the level at which you are doing research, whether it is research for your master's thesis or whether it is research for your bachelor's degree or whether it is for your PhD. It all depends upon that and depends upon the coverage. How much data are you collecting? What is the scope of your study? If your scope of your study is very broad, then you can have more hypotheses. But if the scope of your study is not very broad, then it is suggestible to limit your hypothesis to not more than 5. Below 5 is what is advisable. So more than 4 hypotheses for testing a research is advisable only when the scope of your study is broad. If the scope of your study is not very broad, for example, my own thesis when I did brand positioning is very niche because it's a part of it's a part of bigger marketing communications which are there. I have not taken marketing communication as such, but I've only chosen small brand positioning, which is a very focused narrow topic which is there. So when you have such things, then it is always advisable to have few hypotheses and do more concentrative research on that rather than having many hypotheses and deviating from your original research idea. For a master's thesis, not more than 5 hypotheses is what is advisable. Similarly, people also have confusion with respect to number of sample units that you can have. There's no magical number on number of sample units that you have got. It all depends upon your population. Some of the sampling tables are available like Rexy Morgan table, Cochrane's Q, where you can find out how many sample units can you have for the purpose of your study. So Rexy Morgan is the most widely used sampling table which is available to choose the number of sample units, which says for every 10 million you can have 384 as your number of sample, which is advisable sample based on which you can draw valid conclusions. Again, if your population is very small, as I was telling about accessibility of cancer patients during COVID to healthcare, then my sample is only 10,000 or 5,000 patients. Then out of that 5,000, I can choose only 50. And then I can say that 50 is a justifiable sample which can be there. And also depends upon the sampling technique that you are adopting, that you have to choose your sample which is there. So I hope the purpose for which we gathered for today's session is from my end, I have done it. In case there are any other questions that are there apart from what I have already answered during the session itself in the chat box which were coming. While I was talking, I guess that I have answered most of them. There was a question related to recommending moving away from Cronbach. I have answered that. Somebody was asking related to parametric test applied on sample. I've answered. Mixing of different types of scales. Yes, you can mix more than 4 hypotheses. Are there any other questions apart from the ones which were there in the chat? You know, when there's the silence among the audience, we basically say it is clear or it is all clear. So I am just going with the first assumption that it was clear, whatever I have shared today. The presentation is with the organizers, so you can have a copy from them. Venolia, is there something else that you want me to add to the presentation, because you gave me 1 hour and I guess I'm up with the time. My email, sure.
Okay, I believe that there are no more questions. Or maybe if there's anyone in the audience who would like to unmute themselves and state their questions, maybe this is the time where we give you the platform to ask yourself, to ask your question. You can just unmute yourself and ask directly to our guest speaker. Thank you.
Somebody wants to know if I would be presenting on Smart PLS. Smart PLS would not be, 1 hour wouldn't be sufficient for any PLS, any SM. It would at least 2-3 days or probably 3 hours session and all for that particular Smart PLS, because audiences also need to have a hands-on practice, not simply listening to what is being told, but hands-on is also necessary. So therefore, if something is organized on structured equation modeling, I would suggest that simultaneous hands-on is also available to the participants. That will be more efficient.
Okay, I can see that Abdi Hassan is off mic. Yes, Abdi Hassan, I can see you unmuted yourself. You can ask your question. Okay, I'm now going to give the platform to Naimega. Naimega, I can see you on, you unmuted yourself. You can ask your question if you were about to ask a question. Okay. I believe there are no more questions. Dr. Nazia, would you like to maybe give just a comment about the whole presentation? Maybe just a brief summary of your presentation as in a comment. Dr. Nazia, are you there? You can unmute yourself. You unmute.
You can inform her to unmute her microphone.
Okay, she just wrote in the chat box that I should give her a minute.
Yeah,
Okay.
Vinolia, there was a call from Azan at my end, so that's why I was holding back. So in today's presentation, what we had done is we spoke about in the beginning, we spoke about what is data, what is variable, and from there we proceeded towards understanding the different types of variables which are there, qualitative and quantitative. We spoke about the variables which are independent and dependent, and how these variables need to be analyzed depending upon knowledge related to population and sample that you have got, any technique that you can use to do analysis of those variables. So in case the researchers have got knowledge about their population, then they can make use of the parametric tests which are there. If they do not have knowledge about the population characteristics, then they can make use of the non-parametric tests which are there. Then we spoke about, for these data analysis that we will be doing, it is very essential that we have collected the data on the right lines. So therefore, there are some measurement scales which are available for us to collect data, so that depending upon what data we have collected, we will have to apply the relevant technique depending on data that we have collected. So the four types of scales: nominal, ordinal, ratio, and interval we discussed. And then based on knowledge about the population, different types of parametric and non-parametric tests which are applicable. These parametric and non-parametric can be used together in the same research by the researchers, depending upon any characteristic being known to them or not being known to them, based upon. So there's no hard and fast rule that a researcher will have to use only parametric tests or only non-parametric tests or only a type of a scale in a questionnaire or not use other types of scale. It all depends upon the research objective. It all depends upon the hypothesis and the purpose of the study that is there, that the that respective tools have to be used. And then depending upon what are you intending to do with the data, depending on that, the different types of data technique analysis tools are available to you. In case your intention is to know relationships, then you can do correlation. If your intention is to know impact analysis, then you can do regression. If your intention is to find out differences and variations, then you can do a chi-square. In case your intention is related to understanding linear relationships which are there between many variables and the outcome variable, then you can run a structured equation model, and then you can at once know the relationships that all these variables have got on the outcome variable. So it all depends upon what type of data you have and what type of an end are you going to meet through your research. Based on that, you will have to apply the data analysis tools and techniques that are available to you. It all also some of the data which is quantifiable, you can use quantitative techniques directly. Some data which is not directly collected through quantitative methods or not directly quantifiable or measurable in units, then for such data you will have to convert the data into quantitative form by applying a particular scale and then collect the data. So some data which is purely qualitative in nature, then you will have to run you make use of a software like NVivo to run your qualitative data analysis. So as you know, I was giving example with the help of an Indian fable of an elephant and blind men, that there is no universal applicability of a particular test for a particular type of a data. It all depends upon what is the purpose of your study. It all depends upon how you have collected the data, what scales you have used. Based on that, the data techniques will be different from for different types of scholars who are doing research on similar topics. Also, you can do different types of data analysis which can be used. That was what we covered during this 1 hour session that we have had today. And there were some questions also which were raised in the audiences related to reliability, related to measurement scales, related to knowledge about parameters of the population and applicability of non-parametric tests, related to number of hypotheses that can be used, number of sample units that can be used for a particular research. I hope I have answered those also during the course of my discussion. I thank the Institute for organizing this and giving me an opportunity to associate. I thank Ravikant for being a mediator and liaison between me and between the Institute there.
You're most welcome, Nazia. Nice. Yeah, thank you very much.
Participants who are there, thanks for sparing your time. I hope that the purpose that we have gathered today is being fulfilled, and I have not defeated the purpose or indulged in any, some people feel that I could have done something better. So the opportunity cost, I guess, is little lesser attending this session rather than doing something else.
I just want to say thank you so much, Dr. Nazia. You did a very great job, and I applaud you personally for that. We had such an amazing audience within us, and I just want to say, even in the future, please feel free to maybe work with us and share your knowledge as well. Without any waste of time, I think that time has exhausted. We have pushed to this far, and for those who were requesting for the link for this session, we do have a recording. As you can see, we are live on Facebook. So I would like to say to everyone that if you want a recording link, you can just email me on my email address. You can just email me your request, and then I will share with you the links and so on. And for those who don't know us on social media, we are African Institute for Supply Chain Research. We are on LinkedIn, Facebook, Twitter, and YouTube as well. If you have YouTube, you can just access all of our recordings on YouTube and so on. Without any waste of time, I would like to thank Dr. Ravikant for allowing me an opportunity to engage with Dr. Nazia to bring her into the session to make this wonderful presentation for us. I would like to thank you so much, Dr. Ravikant. And I know
Thank you, Vinali, ma'am.
Yes, I know I can always rely on you in terms of referrals. And thank you for doing that for me. Thank you. And yes, as for our audience, I just want to thank you for availing yourself. And the recordings are available and the slides. So without wasting your time any further, I would like to dismiss the session and have yourselves a good evening.
Thank you.
Bye to everyone.
Thank you.
Thanks.
Bye.
Bye.
Bye.